शेयर को पठाइए ऑन कला पर पठा हुआ
हाँ एठू आप जहाँ कहे ऑन रिकॉर्ड आस
चेंज कर
हाँ हाँ यहाँ एस्केप करदे तापर ये चूज करदे नॉर्मल यहाँ जमी चलिए चलो खाली तांको देखिए जे आपन एटा मिनिमाइज कले बोली ये जमी टिके पर देखिए देखिए एटा मिनिमाइज कले बोली
यूज करेगा
এটা তো বেটা চলবে ওবিএসটা এই যে এই ট্যাবটা কাটিয়ে এটা কাটিয়ে সহজ এটা আসলে
মানে কটে গেছে
ଆପଣଙ୍କଟା ଏଇ ଉପର କମେଣ୍ଟ ଆସିବ
ବଡ଼ ହେବନି
ହଁ ନା ଆପଣ ଫୁଲ୍ ସ୍କ୍ରିନ୍ କଲେ ଏଇଟା ଭିଡ଼ିଓଟା ଆସିବ ବଡ଼ ହେଇକି ଏଇଟା ଖାଲି ଆପଣ ପ୍ରିଭ୍ୟୁ ପାଇଁ ଗୋଟେ
ଆଚ୍ଛା ଷ୍ଟ୍ରିମିଂ ହେଇନି କି
ଚଳୁଛି ସାର୍ କିନ୍ତୁ ଲିଙ୍କଟା ଦିଆହେଇନି ତ କଲେ
আসলে
डॉक्टर सूर्या को फिजियोलॉजी सर ये जो तुमको लिंक पठेछि देखो ए फिजियोलॉजी क्लास गोटे तुम अच्छी ना डॉक्टर विराजमान सर ने जो मेल रिप्रोडक्टिव सिस्टम सकाल जमी यूट्यूब लाइव होता सेमती यूट्यूब लिंक गोटे पठेछि तुम ग्रुप रे शेयर कर दो को पिला को जॉइन कर देबो मैंने देखिकी पढ़ाई तो टोटाल 17 व्यू
ये आस
देखिए के जन देखो एससीपी जो शेयर करेंगे एससीपी जो लिंक शेयर करेंगे टिके लैग रहे टिके माने 10 सेकंड लैग रहबो माने अपन एबे कहले ताको टिके डिले सुबह आज अपन कमेंट सेक्शन देखियो फोन रे भी से जो लिंक रे आसले क्या जान कर ले चले चलो स्टार्ट कर सर टिके फर्स्ट अपन लिंक पठे दीजिए यदि आप फोन रे खोलिक रखिए आप देखि पारे कमेंट सेक्शन के कोन जे कहै छै कि
एटा आसो
कि आप एबे तो एरा खोल देबे ना माने पीपीटीटा आपन को खाली एटा ही देखिबो आपन जहा कहबे तांको देखिबो बट आपन जानि परिबे जे केमते देखा कि कोन चेंज होला फोन रे खोल देबे केते लोक अछि एटा भी देखियो आपण तेरा देखो जे मतलब कोई तो सरले से कर दे क्लोज कर दे
एबे लाइव अछि
आपण प्रेजेंटेशन मोड करले आसी पिला को देखियो टिके लैग रहबो सर 10 सेकंड में देखम आसी
Hello, good afternoon students. Hope you are having a nice time at your homes. The summer is too hot right now. Okay. Very well. So today as we're supposed to carry on with our reproductive system classes, and if you remember in the last class we talked about how the differentiation of sex occur, and today we will talk about more specific about the male reproductive system. So if you recall from your last class, we have already discussed about how the undifferentiated gonads in the presence of your Y chromosomes having a cervix gene, which is your testis determining factor, gives rise to your gonadal testis, and this testis finally gives rise to your testosterone as well as secretion of your Mullerian inhibiting substance. Now, as you know, the Mullerian inhibiting substance inhibits the Mullerian system and allows the Wolfram duct to develop and differentiate it into the male ductal system along with the bladder and the urethra. So this already we have talked about, and you can see in the finally the developed system you can see the Wolfram duct finally gives rise to your testes as well as your epididymis, vas deferens, seminal vesicles, prostate, urethra and the penis. So together with the help of testosterone, your infantile internal and external genitalia are developed, and today we are going to talk about, basing on that background understanding, we'll talk about today the male reproductive system. So today's objectives will be to identify the structures and the major organs of the male reproductive system and describe their functions, as well as we'll also talk about tracing the pathway of sperms cells in the organ system. So we'll also talk about the spermatogenesis as well. So now if you see this structure, I think you are all aware of this structure because from your +2 or your NCERT level also you have seen this, you have covered this up, so just a brushing up of your memories. So you can see we have divided the male reproductive system into glands and the ducts of testis as well as your supporting structures. So if you talk about the glands, we have got a pair of testes, a pair of seminal vesicles, prostate and a pair of bulbourethral glands or the corpus glands. Now if you talk about the ductal system, now the testis where this your spermatogenesis occur or the sperm production occurs, which has been poured into the epididymis. So we have got on the both side, we have got a pair of epididymis, a pair of your vas deferens, and then a pair of your ejaculatory duct, and finally all the secretions are being finally poured down into the urethra. And if you talk about the supporting structures, we also have got internally, if you talk about, you have got a pair of spermatic cords which holds the structure within the scrotum, as well as externally if you talk about, you have got a pair of scrotum and a penis. So today's discussion will be surrounding your testes. So if you take a cross-sectional lateral section of your testes, you can see they have got multiple lobules, okay, which is filled with your tubular structure, which is around 1.5 meter long, which are known as your seminiferous tubules. Almost around your 900 seminiferous tubules are present, which are placed within the lobules, okay. And then if you see, this is the seminiferous tubules where your actual sperm production occur or the spermatogenesis occur, which are finally poured into the retitestes, and then from the retitestes it has been collected into the through the efferent ductules into the epididymis, and then from the epididymis it passes on to the vas deferens, and then through the ejaculatory duct into the urethra. So this is the overall structure of the testis and how it looks and how it is connected. Now if you go to the further detailed structure, if you take this section of a seminiferous tubule, it looks somewhat like this. If you see in the diagram A, you can see this is a picture of a single seminiferous tubule, round structure, and the other you can say your half moon shaped structures, they are the different seminiferous tubules, and in between the seminiferous tubules you can see the interstitial space where you have got special cells which are known as interstitial cells of Leydig. And more specifically now we will talk about the seminiferous tubule. If you see the seminiferous tubule, it is rounded structure with a basal lamina around, and you can see a lot of cells are there which are mostly your spermatogonium and different stages of spermatogenesis leading to finally the production of your sperm. So you can see different stages here, and if you take a more magnified view of it, you can see here at the basal layer, close to the basal lamina, you can see the spermatogonium, and then if you go further down, you can see the primary spermatocytes, secondary spermatocytes, and towards the central lumen you can find the final spermatids and the which are being released as sperm. So this is how you can see from the base towards the apical area or the lumen, the maturation goes on. And if you take a larger view of this, you can see again the basal lamina, and along with that you can see this long cells which are the Sertoli cells in between, and along with the Sertoli cells you can see the speciality of the Sertoli cells are that they are the cells with having a larger cytoplasm which is hanging around, and the junction between the each Sertoli cells, they make a tight junction which is known as your brain-testis barrier. And the more interesting is this 3D structure. If you try to understand, the Sertoli cells are like covering up, and in between them the cells of maturation from the spermatogonia to the mature sperm, they pass on and they differentiate on the way through. So this is a view where you can, if you try to understand, you can understand, and this is how the divisions are going on from the spermatogonium finally to the spermatozoa, how they pass on from the different stages of development all through the from the basal lamina towards the central lumen, embedded within the Sertoli cells. So one more interesting term is here which we passed through, and you must know the importance of this blood-testis barrier. So if you go back to the previous slide, you can see what is the blood-testis barrier. Here you can see the two Sertoli cells and the tight junctions made by the Sertoli cells, they form the blood-testis barrier. Now there is a tight junction between the Sertoli cells, particularly near the basal lamina, and this is the main reason which prevents the large molecules like your steroids to pass through. As you know, the steroids are hazardous for the spermatogenesis, so that is why this blood-testis barrier helps or prevents the steroid to pass into the area of spermatogenesis. So the major functions of the blood-testis barrier is mostly to maintain the composition of fluid in the tubular lumen. Now that is inside the seminiferous tubule. If you see the fluid composition, it is little bit different from the extracellular fluid, which is here you can see they have got very little protein, less glucose, but rich in your steroids like androgen, estrogen, potassium, inositol, and glutamic and aspartic acids, which is important for the maintenance of this spermatogenesis. So they provide the nutrition and the support for the growth and differentiation from the spermatogonia to the sperm. The second most important function is that it protects the germ cell from the blood-borne noxious agents. Now if you see the outside of the basal lamina, you can see in between the, as I told you, in between the seminiferous tubules in the interstitial space, you have got your Leydig cells, as well as all the blood vessels are also passing through those areas. So the blood capillaries are in very close contact with the basal lamina, but because of the blood-testis barrier, the noxious agent that is passing on through the blood or the different drugs that is passing on through the capillaries are not easily allowed to pass through into the area of spermatogenesis, which can hamper your spermatogenesis and make the person infertile. As well as it also prevents the autoimmunity due to antigenic products of the germ cell. What does it mean? It means that whenever there is differentiation, there is breaking down of the larger molecules of your spermatogonium on the way through the development towards the spermatids, and these are actually antigenic structures, so which can give rise to formation of autoantibodies if they come in contact with the blood. So that is why, because of the blood-testis barrier, the autoimmunity is prevented, and this gives rise to a safe environment for the spermatogenesis. So more specifically talk about Sertoli cells. We have already discussed about how the Sertoli cells look like. So they are the cells having a hanging or larger cytoplasm, and which secretes your androgen binding protein. So this helps in the binding of the receptors they have receptors for binding the androgens, most particularly like your testosterone, as well as it secretes your inhibin and mullerian inhibiting substance. So mullerian inhibiting substance we have already expressed how it helps in the development of the ductal system, as well as inhibin, which gives a negative feedback to the higher centers like your anterior pituitary and the hypothalamus, in order not to release more of your LH and FSH. As well as it expresses the aromatase enzyme, which actually helps in the conversion of your testosterone to estrogen. As well as it provides glycogen, which nourishes the germ cell, also provides the proteolytic enzymes, which helps in the releasing of mature sperm, because as we already mentioned that whatever the development occurs from the spermatogonad to the spermatid occurs along with the attachment with the Sertoli cells, and finally the detachment of the the spermatids into the sperm, and finally the sperm being released from the Sertoli cells, which is otherwise known as spermation, takes place due to this proteolytic enzymes which is being secreted from the Sertoli cells. As well as we also talked about the various particles that are being released, broken down during the differentiation, are also known as the residual bodies, which are being phagocytosed by the Sertoli cells. So they also have a phagocytic function. And if we talk about the Leydig cells are also known as interstitial cells of Leydig, which are abundant in the early fetal life, and then again it decreases till the puberty, and once the person attains the puberty again it increases, and almost the secretion will be around 7 to 8 milligram of testosterone per day, which is directly under the influence of luteinizing hormone, or LH, most commonly termed as LH, which is being secreted from the anterior pituitary. So Leydig cells are directly under the influence of your LH, which facilitates the growth of your accessory sex organs as well as your development of your secondary sexual characteristics. So the Sertoli cells and your Leydig cells, each one are the short nodes. Now coming to the most important part, that is your spermatogenesis. So as we have already talked about how the germ cell passes on to the infantile gonads, and how the germ cell gets differentiated into the sperm. So before entering into the puberty, the germ cell undergoes, the primordial germ cells undergoes multiple mitosis, and you can see up to 12 to 14 years, the most important feature here is that the primordial germ cell passing through your basal lamina and then entering into the seminiferous tubule. That is the first step, and once it enters into the seminiferous tubule, then it differentiates and enlarges, swells up, and gets the name of spermatogonia, and it undergoes mitotic division, giving rise to two daughter cells with the same diploid number of chromosomes, and then it goes into the stage where it becomes a primary spermatocyte. The primary spermatocyte is nothing but one of the daughter cell getting swollen up and increases in size, and this primary spermatocytes, this undergoes the first meiotic division, giving rise to secondary spermatocyte. And here you can see the secondary spermatocyte again passes on into the second meiotic division, where it gives rise to 4 daughter cells having a haploid number of chromosomes, which finally gets differentiated into a mature sperm. So here you can see the two terms are there, like mitosis you understood about the meiosis 1 and 2, and then the spermiogenesis. Now spermiogenesis is development of spermatids from the secondary spermatocytes to spermatids. But once they have become the spermatids and develop into the mature sperm, they are still stuck to the Sertoli cells. Now spermiation is a process which actually helps the release of this mature sperm from the Sertoli cells into the lumen of the seminiferous tubules. So once it is released, how it looks, you can see this is the structure of the mature sperm, having a head with a cap, which is an acrosomal cap, and it contains a vacuole as well as the anterior head cap, posterior head cap. So this head is the most important structure, which contains your proteolytic enzymes and the hyaluronic acids, as well as you can see the straight neck, which has got multiple microtubules, okay, and the multiple mitochondria are present. So this is the most active zone, and then finally the tail. So this is the normal sperm, how they look like, and this acrosome is most important because of the enzymes that they carry, and this, due to the proteolytic enzymes, they are capable of or have the potential to penetrate the ovum by fusing or releasing the enzymes which dilutes the cell membrane of the ovum and allows the sperm to enter. But then when this sperm is released, actually it is released into the, as we talked about, from the seminiferous into the your cavity, into the lumen, seminiferous tubule lumen, and into the lumen when they are released, they are actually non-motile. That means they are non-functional. At this stage, even if they are allowed to fuse with the ovum, it won't work out. There won't be any fertilization. So they don't have the ability to fertilize. And they are stored in the epididymis for a long time, where they move very slowly through the ciliary push, where they are stored and protected for a longer time, and even for the months they can be stored here. During this time, actually, they regains the maturation and the ability to mobilize. And once they attain the mobility, once they are entered into the passes through the ejaculation and passes into the, or when they are injected into the female genital tract, where they get the capacitation or the actual the strength to fertilize the ovum. So what are the roles of the accessory sex organs? So as you know, the semen not only contains your sperm but also the fluid along with it. So the fluid that it contains along with it are mostly secreted from the seminal vesicles, like your fructose, prostaglandin, which actually helps in the mobility and transport, as well as your fibrinogen, citric acid, ascorbic acid, and your vitamin B. Along with your, if you see the prostatic secretion, they are mostly in alkaline fluid, as well as they contain your fibrinolysin. As in the last class I mentioned about, when they are secreted into the female genital tract, they are in the coagulum state, or whenever the ejaculatory, if you see, they are in the coagulum state because of the presence of fibrinogen. But after, if you allow it to stay in the room temperature for within 30 to 40 minutes, they get lysis because of the presence of secretion from the prostate, which contains your fibrinolysin. As well as the semen also contains your mucous secretions, which are mostly coming from the urethral glands. So what are the factors that actually help in the spermatogenesis? The first and foremost is your testosterone, as we all know. Along with that, the luteinizing hormone, which is being secreted from the anterior pituitary, which actually initiates the spermatogenesis, mostly through the stimulating your Leydig cells to release your testosterone, and which helps in the spermatogenesis, as well as the follicle stimulating hormone also helps in the release of spermatids to final maturation of the spermatids to the sperm, which is mostly under the influence of the follicle stimulating hormone, as well as estrogen, growth hormone, and temperature. If you talk about the temperature, as we all know, the testes need to have a temperature at least 2 to 3 degree less than that of the core body temperature. So that is why the testis has been kept outside the body. So the temperature is very important. If they are exposed to higher temperature, they may hamper the testosterone. They may hamper the fertility or the production of sperm or the spermatogenesis. That is why you may see the people who are working in the blast furnace or who are exposed to constant heat during their work nature. In long term, you can find that they undergo infertility because of the exposure to high temperature for longer duration. So that's all for the first class we talked about, and as I have already mentioned about the semen and the semen analysis, so I won't talk about that in details right now. So we'll finish the first class here, and I've got one more slide for you, which is the steroidogenesis or the production of steroid. So just wait for 1 minute. Okay, so today we'll try to cover up the whole of the male reproductive system. So I've tried to minimize the slides so that you can get the things that is at most required for you, not going into more of the details. So now we'll try to cover up the steroidogenesis or the endocrine function of the testes. So as you know, the androgens, which is most commonly termed, are the hormones which are mostly secreted from the testes, as well as the hormones which are also secreted from the skin and also from the adrenal glands. But then when we say androgens, the hormones which are mostly responsible for the maleness in the male. The most common hormones are like your testosterone, androstenedione, and dihydrotestosterone. But for all practical purposes, we mostly use the testosterone because that is the maximum production. And most commonly, when we think of androgens, we think mostly of the testosterone. So if you talk about the biosynthesis of the testosterone, it is mostly your carbon-19 structure, which is a steroid, having a hydroxyl group at C17, and this is the hormone mostly under the category of steroid hormones, which are being secreted mostly from the testes. And if you see the substrate from where this production occur or the biosynthesis occur, is mostly from the cholesterol. So if you see here, we're not going into details of it. Now if you see here, it is mostly converted into pregnenolone, and how the pregnenolone can also be take a second different part to give rise to progesterone, as well as how the pregnenolone gives rise to 7-alpha hydroxy pregnenolone, which further gives rise to dehydroepiandrosterone and your androstenedione, and this androstenedione finally gives rise to your testosterone. And in the presence of your 5-alpha reductase, particularly in the target tissues, mostly your testosterone which is being secreted are mostly in the bound form, and once in the bound form they travel in the blood, and once finding the target tissues which contains the 5-alpha reductase, gets the testosterone converted into dihydrotestosterone, which is the most active form. So this is the more detailed structure how this testosterone is being secreted. On the left side of my picture you can see the Leydig cells, and on the right side you can see the Sertoli cells. So as we all know, the Leydig cells are responsible for the secretion of the testosterone because they contain the receptors for the LH hormone which is being secreted from the anterior pituitary. Now once this luteinizing hormone gets attached to the receptor on the membrane of the Leydig cells, and these receptors are mostly your G-coupled protein receptors, which further gives rise to your activation of the adenyl cyclase which is attached to the cell membrane, and this adenyl cyclase catalyzes the formation of your cyclic AMP, and the cyclic AMP finally gives rise to the activation of the protein kinase A. Now these are actually the mechanism of hormones. Like when you talk about the hormones, now if you see here you have got your different receptors at different levels. Like some of the receptors you have got at the cell membrane, some of them have the receptors at the cytosol level, some of them have the receptors at the nuclear membrane level. So depending upon the classification of the receptors, we have got the hormones as well as the receptors, and for individual hormones the receptors are being placed at different levels. So when you taught about the mechanism of hormone reactions, there you can understand the details of which kind of receptors get attached to which kind of hormones. And this protein kinase activation, protein kinase A activation helps in the transcription of mRNA, giving rise to synthesis of your new proteins, which can be your enzymes, which we talked about how the cholesterol gives rise to formation of the testosterone. As well as if you see about this testosterone get transfused poured into the circulation. As you know, all the hormones are being finally poured down into the circulation, and finding the target organ, like here is the target organ is the neighboring Sertoli cells. So there also it passes on. As well as if you see about the mechanism of hormone action of hormone along with the Sertoli cells, here also you can see they have got receptors for the follicle stimulating hormone. So Sertoli cells are under the follicle stimulating hormone influence, whereas your Leydig cells are under the influence of your LH hormone. So here also they get attached to the receptors which are G-coupled protein receptors, and here also the activation of adenyl cyclase, and which catalyzes the cyclic AMP production, and increase in cyclic AMP production, how it gives rise to activation of the protein kinase A, almost similar to that of the activation in the Leydig cells, which finally gives rise to synthesis of new proteins. So what are the new proteins synthesized here? There are the inhibins, androgen binding protein, as well as your aromatase. And this aromatase is the kind of enzyme which actually helps in transformation of the testosterone to estradiol. As well as it also helps in production of your growth factors and other products, which actually helps in the synthesis of new proteins, or promotes the synthesis of new proteins in the Leydig cells. So you can see there is constant talking between the Leydig cells and the Sertoli cells, which actually in coordination with each other talking and helping in production of testosterone as well as controlling the production, and finally helping in the synthesis of or the spermatogenesis. So as we talked about the production of estrogen in males, yes, definitely the estrogen in males are being produced, but through the Sertoli cells only the 20% of that estrogen in males are being produced, but rest of the 80% are being produced through the liver, adipose tissue, as well as skin. Now coming to the testosterone, if you see, once they are produced, you can see the production of the testosterone is mostly has got a diurnal rhythm. That means not all throughout the day the testosterone is secreted, but the peak are mostly in the afternoon and the evening. And you can see also the increase in testosterone secretion hours after walking. And most of the testosterone that are secreted, around 95% of the testosterone, are secreted from the testes, whereas 5% of the testosterone are being produced from the other sides like your adipose tissue, skin and the adrenal cortex. And once they are produced, you can see 97% of the testosterone produced are bound in the plasma through the plasma proteins. Mostly they are bound to the beta globulin, which is otherwise known as the sex hormone binding globulin, and in the bonded form they are transferred to the tissues, where in the presence of your enzyme 5-alpha reductase get transferred into just converted into the dihydrotestosterone, or if they don't find the target tissues, they get degraded finally. And this degradation occurs mostly in the liver, where they get conjugated and finally degraded into your finally they are conjugated and are being released into the gut or passes through the urine, and that is how they are being excreted out of the body. So the most of the action is seen mostly in the prostate gland, and as I told that most of them are in the bound form, and hardly the 2% of the free testosterone are mostly responsible for the mechanism of action in the prostate gland. And in the prostate gland they have the androgen receptors, particularly in the nucleus, and because of the presence of your 5-alpha reductase enzyme, this testosterone is getting converted into dihydrotestosterone, which is the highly potent one, and it gives rise to new protein synthesis through the increased transcription in the nuclear chromatin, finally giving rise to protein synthesis, as well as there are some non-genomic actions are also seen, which stimulates the hepatic microsomal protein synthesis, as well as stimulation of adenyl cyclase enzyme, giving rise to activation of the protein kinase in the prostate gland as well. If you talk about the effects of testosterone, when in the intrauterine state, the testosterone basically helps in the development of the Wolfram duct, that is mostly the male reproductive tracts, as well as the dihydrotestosterone, which helps in the development of the male external genitalia, prostate gland, as well as it helps in the descent of testes by 7 months of the intrauterine life. So this descent of testes is very important, because by 7 months to by the time baby is born, this testis has to be descended down, and if it is undescended, it is known as cryptorchidism, which is a clinical case which further needs surgical descent of the testes, because if it is undescended, it may remain either in the spermatic cord, or else it will remain in the inside the stomach. If it still remains in the stomach, it may not give rise to the production of sperm, because as you know, the core temperature of the abdomen is much higher than that of the scrotum, which may even further lead to differentiation into the tumor, testicular tumor. So that is why the descent of testes is very important in the males, which actually defines the fertility. And after birth, this testosterone decreases or ceases, giving rise to small testes, which is non-functional till they reach the puberty. So at the puberty, by around 10 to 14 years, you can see the first change in the males, that is the enlargement of testes, which is otherwise known as spermachy, which gives rise to spermachy, that is the first production of sperm, and external genitalia growth, as well as your growth in the prostate, as well as along with its secretion, distribution of the body hair, voice, change in voice, that is the cracking of voice, and the body conformation, as well as development of muscles, skin, and the secretions from the sebaceous gland, which is typical of the male. So these are the changes of the, or we can say the secondary sexual characteristics, which is mostly due to the production of the dihydrotestosterone. And apart from the sexual growth, you can also see the other features where this testosterone are helpful, like in the growth of the bone. So mostly your increase in the bone matrix, as well as your calcium retention, it also helps in the increase in RBC. Most of the time we ask you in the orals or practical orals that why males have got more RBCs as compared to females. So here you can see they have got a indirect effect due to the increased metabolic rate. So there is little bit higher production of your RBC. So that is why your RBC content is more as compared to females. As well as your testosterone gives rise to increase in BMR, abdominal visceral fat, as well as it helps in the electrolyte and water balance as well. Apart from that, there are some other features also, like your aggressive behavior, sexual potency, spermatogenesis, maintain reproductive tract throughout the adulthood. Now you must know that once the spermatogenesis begins, it actually remains throughout the lifetime in the males as compared to females, like they attain a menarche and then they attain menopause where it finishes. But in case of male, it maintains throughout the lifetime, but it has been seen that above the age of 60 years it gradually decreases, but still the spermatogenesis continues throughout the lifetime. So how this testosterone are being regulated? This is a picture taken up from your guidance book. So I think all of you must be able to draw this diagram. This is a very schematic diagram. So here you can see from the arcuate nucleus of the hypothalamus how this gonadotropin releasing hormones are being secreted, which stimulates your anterior pituitary to release either your FSH or LH. And you can see that in a cyclical manner, and particularly the LH is being released in a pulsatile manner. That means they are released in the series or pulses. There is no constant or continuous release. So there is a pulsatile release of LH, and you can see how the LH stimulates your Leydig cells of the testes to secrete your testosterone, giving rise to the androgenic effect. As well as the testosterone also gives rise to a negative feedback to the anterior pituitary as well as your hypothalamus to prevent the further secretion of the testosterone. Otherwise there will be constant release of testosterone, so this gives rise to a negative feedback effect. So like we always say that you have a good bike, but if you don't have a braking system in a bike, then it is a, what is it? Is it a good bike or it's a killing machine? Definitely it's a killing machine. So that is why in the bodies is also whatever are being secreted doesn't continuously go on secretion. Rather, they also get a negative feedback which actually controls the secretion of the testosterone and regulating your androgenic effect. Similarly, the anterior pituitary also secretes a follicle stimulating hormone which has a stimulatory effect on the Sertoli cells, which gives rise to spermatogenesis, as well as it secretes the inhibin, which we have seen, which actually helps to get a negative feedback on the anterior pituitary as well as your hypothalamus to inhibit the further secretions of the FSH. So this is how the regulatory mechanism of the FSH and LH goes on. So for the applied aspect, as we talked about andropause, there is no clear cut of andropause, but it has seen that over the years of 60 years, the spermatogenesis decrease. So you can see in the graph, this in the x-axis talks about the duration of trimester. There's the first trimester, second trimester, and third trimester. This is about the intrauterine life till the birth. So you can see here, during the second trimester, there is increase in the plasma testosterone, which is secreted from the gonadal testis or the infantile testis, and just immediately after the birth there is decrease of testosterone after some time. The initial stage there will be slight spike, and then again it decreases and remain quiescent till the attaining of the pubertal stage. So once they attain the puberty, again there is increase in the testosterone, plasma testosterone, and here you can see this testosterone actually helps in the production of the sperm as well as gives rise to secondary sexual characteristics. And along with that, the blue line, if you see, it is the sperm production, which goes on and takes the peak throughout your 40 to 60 years, you can see, and after the 60 years you can see it is gradually decreasing. But if you see it doesn't touch the baseline, that means it doesn't come to zero. So there is no andropause. So this is all for the day, and I think for the today the questions will be mostly your spermatogenesis. So please prepare with the spermatogenesis, which is your long question, as well as you can also prepare with the Leydig cells and the Sertoli cells, which may come as a short note. So thank you all.